Hello everyone, I am Baran Taşdemir and I am a PhD student at Hacettepe University Computer Engineering Department. And I'm here to represent my team, Biological Data Science Lab, and I will present FlowProt, classifier-guided flow matching for targeted protein backbone generation in the de novo DNA methyltransferase family. So I want to start with a short introduction on protein design. Proteins are critical to life's functions, and their function is directly linked to their 3D structure, which emerges from the sequence. Designing proteins with novel or enhanced functions can lead to powerful therapeutics or new biomolecular tools. However, doing this from de novo has some difficulties. First of all, the sequence space is huge. For example, a 200 residue protein has 20 over 200 possible sequences. So experimentally exploring this space is both slow and costly. That's why there is a need for data-driven computational models that can help us efficiently generate proteins that are both stable and functional. In the protein design problem, we typically aim for two key outcomes: structural stability and functional relevance. So a valid design must ensure a proper folding and function. And many generative models produced today focus on the structure, but function also tends to be overlooked. In this slide, we show a representative work on the literature, RF diffusion. It's a diffusion-based approach from Baker Lab that inspired many recent advances. Diffusion models are a class of generative models that simulate the data generation by processing a data distribution into a desired complex data distribution by using some sequence of operations. Diffusion models are trained to learn a denoising function. So in training, a clean sample is corrupted by a noising function at each step until it reaches a total corrupted form. And then the model learns a denoising function that will lead to the clean sample from that noisy sample. So diffusion models add noise and learn to revert it to generate structure. They are easy to train, but their inference is expensive. Flow matching, on the other hand, learns the mapping from noise to data distribution. So it makes the training process harder, but it provides a faster inference. So in FlowProt, we are using the flow matching models advantage on the inference to generate efficient backbone generation. Our goal is to build a general purpose generative model that can create protein backbones tailored to a function fast and accurately. So we are using flow matching strategy to utilize its speed on the sampling. And we test this idea on a challenging case, de novo DNMT family proteins. DNA methyltransferase family of enzymes catalyze the transfer of a methyl group to DNA. So DNA methylation serves a wide variety of biological functions. DNMT3 mediated DNA methylation plays a leading role in the execution of many critical processes such as regulation of transcription, embryonic development, or genome stability. Because of its catalytic role in the inhibition of target gene transcription, DNMTs plays a significant role in maintaining a chromosomal homeostasis. Overexpression is observed in cancer. It silences tumor suppressor genes. Defective DNMTs cause imbalance in DNA or histone modification, this resulting in chromatin remodeling, genomic instability, and gene inactivation. So accordingly, designing a new enzyme with the NMT function has the potential to be applied in both therapeutical and biotechnological fields. So therefore, we aim to design and test de novo DNMTs. We selected this family because it is well studied and the molecular mechanisms are well known. But as a goal, we are designing models that can work with any domain or any function. So FlowProt is a generative model that outputs 3D backbone structures residue by residue. Unlike other general purpose flow models or generative models, we steer the generation using a classifier to target specific domains like, in this case, the NMT. We use a frame-based representation derived from frame-diff. Each residue's position is represented by its alpha carbons coordinates in Euclidean space. And orientation is encoded by using Gram-Schmidt orthonormalization, allowing us to represent the local geometry. And the model learns to predict the translations and rotations that will carry residues to their final position. So to inject functional knowledge, we use a binary classifier trained to distinguish the NMTs from others. So it is inspired from diffusion models classifier guidance. During inference, we use the gradient of the classifier score to adjust generation steps, pushing output towards the NMT-like structures. For training, we used over 35K DNMT domain proteins for the generative model and 65K proteins for the classifier model. The classifier training set includes both positive DNMT samples and negative non-DNMT samples. To add a challenge to the classifier, we also construct half of our negative samples from methyltransfarase proteins, which are very similar to the NMT proteins, but not belong to the NMT family. And to validate our designs, we use a evaluation pipeline. It has 3 steps. For each backbone that our model is generated, we generate a sequence from these backbones by using protein MPNN and predict the folded structure by using ESM fold. And we compare with the original backbone using self-consistency, RMSD, and TM scores. To evaluate the functionality of our designs, we use progress similarity score between human DNMT3A. So progress is a tool that can calculate similarity between proteins by using learned latent representation of protein structures. We tested multiple sequence lengths. For each sequence length, we generated 10 samples. By using the evaluation pipeline, we generated 9 folds for each sample. And we present average scores for the top 5 samples per sequence length in the table. So mid-range lengths like 128 to 286 residues performed best, producing structurally stable and functionally relevant proteins with low RMST and PLDT scores. So I want to talk about each metric one by one. Self-consistency RMST and self-consistency TM scores are structural scores. So RMST score computes the average distance between alpha carbons in the original and folded structure. It is a lower is better metric. And we ask the question, did the design sequence fold the way that we wanted to? So in the plot, we can see that for all sequence lengths except 512, our model can generate designable backbones. So we can see very low RMSD scores for each sequence length. So this means that our model can generate designable proteins that has very low RMSD scores. And self-consistency TM score measures the topological similarity between 2 structures. It is a global score, and SCTM is more sensitive to global shape than local shifts. For this metric, equals to 1 means perfect match. So we can see the same trends as before, as previous plots. So our model can generate backbones that are designable in terms of self-consistency TM. So there we can see very high TM scores here, except 512. So for proteins with larger sequence over 286, we think that our model cannot learn due to lack of data samples with large sequence lengths. In the training set, the average length of sequences are about 300. So it is an expected behavior. And also for this DNMT case, the reference protein DNMT3A has 286 residues. So we expect that the model cannot generalize over larger protein sequences. In this plot, we observe the progress similarity score that evaluates our design similarity to the reference DNMT3A protein. We can see that at the target sequence length 286, we can generate proteins with very high similarity, 0.89. This means that the probability of the functional relevance is higher for our generated proteins. Even with smaller sequence lengths, our model can generate very similar samples. So here. As the next step, our collaborators that are experts on the field tested our models or top models using Chai-1 to predict SAM binding. We used two new metrics to evaluate. So first one is PTM. PTM is the predicted TM score that showed the confidence over the predicted structure. And the other one is IPTM. IPTM score is inference predicted TM score. Shows the confidence on the interaction. We see high IPTM and PTM scores on some samples, suggesting potential to form stable SAM bound pockets. One model even reaches an IPTM score of 0.76. So these top designs will be taken further with MD simulations. And also we tested the DNA binding capabilities of our samples. Here's an example done by our collaborators again. They are using Chai-1. We tested if the samples can bind with DNA, and we found that our samples can bind. So backbone 37 on the left can bind with DNA with 0.72 IPTM score, and backbone 38 on the right one can bind with DNA with 0.41 IPTM score. Both samples can bind to DNA. However, while the confidence for backbone 37 is high, the confidence on backbone 38 is low. So these two samples can also bind with SAM with IPTM scores 0.37 and 0.76. This means that for backbone 37, while the Chai-1 can bind SAM with lower confidence, it can bind DNA with a very high confidence. It is the opposite for the backbone 38. These are only preliminary results, but the tests are showing the potential for DNA binding. So we are currently working on testing more samples to analyze. So far, we present preliminary analysis done by our collaborators. The reason that we choose this kind of evaluation strategy is that we are trying to find strong candidates from a populated sample group. As next steps, we are planning to send these samples to the manual testing, and next we plan to test stability with molecular dynamics and test the DNA binding capability of our designs. And we are also preparing a public release of FlowProt with a web tool and public weights for other researchers to use. To wrap up, FlowProt is the first flow matching model guided by a classifier to generate domain-specific protein backbones. It is efficient, function-aware, and open to generalization. We use the NMT domain as test case to demonstrate our model's performance, but we designed the model as generalizable over other protein families or functionalities. And our project is open source in our GitHub repository. You can try to use it if you are familiar with Python coding. This work I presented is a part of a large project called ProtGen. So this is a collaborative project between 3 universities, which we lead as Hacettepe University Biological Data Science Lab, led by Professor Tunca Doğan. I'm also a member of this lab responsible for the AI modeling. Since I don't have the expertise in the chemistry and biomedicine fields, we have expert collaborators, Dr. Ezgi Karaca and her team from İzmir Biomedicine and Genome Center, who is dealing with DNMT-related physics-based analysis of the structure, and Dr. Abdurrahman Olaç from Gazi University Pharmaceutical Chemistry Department, who is conducting protein ligand interaction experiments. And this is our team. We are working on different projects as a team. So I want to thank them all for their contributions. If you are interested in our works, we have different talks and poster presentations in conference. So before closing, this is our talks and posters. Thank you for your attention.
Thanks for a great talk. Does anyone have any questions?
Hi, that was a very nice talk. So, have you looked at this paper RERD, which is from Sergey Levin and Aviv Levin's group? It's kind of similar. They also do reward guided diffusion, where they use it in the denoising. And I was curious how your approach would compare to theirs. So they also do sequence design, and then they code it using structure-based models.
So can you repeat the group's names?
The method is called RERD, and the authors are Sergey Levin at UC Berkeley and Aviv Regev, who's at Genentech.
No, I haven't heard, but I will definitely check
Yeah,
after.
certainly worth looking at.
Yeah, thank you so much.
Anyone else have questions?
Hi, thank you for the great talk. Do you have an estimate for the required number of structures for generalization of other protein groups?
Yeah, actually, we are using 65K proteins, and I believe that it is enough to generalize over some kind of protein geometry. But I believe that we need more samples to generalize over some specific domain or function.
Okay, thank you very much.
You have time for a few more questions?
Hello, thank you for such a nice talk. I was wondering, when you do your diffusions to bind DNA, and then I think you generate sequences with protein and PNN,
Yeah.
do you condition the PNN somehow to make the DNA explicit somehow in the design, or just let it be?
No, we don't have any conditioning on the evolution pipeline, so we are using only raw backbones and using protein and PNN to produce like 3 or 4 sequences, and then forwarding them to ESM fold without any conditioning on binding or any other thing.
Interesting. Thank you.
All right. Anyone else? Okay. Now we're going to go to the next speaker. So we're going to invite Remo Bros.